Good morning. I'm Devasena. In this lecture, we are going to discuss about respiratory measurement. In detail, we are going to discuss about the function of lungs and various lung volumes and capacities. Finally, we will discuss about the instrument spirometer. Respiratory measurement. The exchanges of gas in any biological process is termed as respiration. Here is a representation of respiration. The process of taking in oxygen from the environment, transporting the oxygen to the cells, removing the carbon dioxide from the cells, and exhausting this waste product into the atmosphere is called respiration. Respiratory system process is shown here. The exchange of gases takes place through the membrane of the pulmonary alveolus, which always contains air. Oxygen is absorbed from the air into the blood capillaries, and the action of the heart circulates it through all the tissues in the body. At the same time, carbon dioxide is transmitted from the blood capillaries into the alveoli and then expelled through the bronchi and the upper respiratory tract. In the human body, the respiration can be classified as internal respiration, that is, the exchange of gases between the bloodstream and nearby cells inside the body is called internal respiration, and the next one is external respiration. The exchange of gases between the lungs and the bloodstream inside the body is called external respiration. Breathing consists of two phases. They are inspiration or inhalation. The process of taking in air is known as inspiration. The intake of air into the lungs through expansion of chest volume. And the next phase is expiration, the process of blowing out air. This expiration is also known as exhalation, the expulsion of air from the lungs through contraction of the chest volume. This is represented here. Inhalation, exhalation representation is shown here. During inhalation, air enters into the windpipe. Due to that, rib moves out. The volume of thorax increased. Because of this activity, the diaphragm gets contracted. Now, during the exhalation, air leaves. How the air leaves? Diaphragm relaxed and arched upwards. Due to this, the volume of thorax decreased. Rib moves back and air leaves from the nose. This process is represented here. Respiratory system parts is shown here. The respiratory system is divided into two parts. They are upper respiratory parts and lower respiratory parts. The upper respiratory parts includes the nose, mouth, and the beginning of the trachea. That is the section that takes air in and lets it out. And the lower respiratory part includes the trachea, the bronchi, the bronchioli, and the lungs. The organs of the lower respiratory tract are located in the chest cavity. They are delineated and protected by the rib cage, the chest bone, and the muscles between the ribs and diaphragms. Lung volumes and capacities. The lung volume and capacities are determined to assess the state or the condition of the breathing mechanism. Here is a representation of lungs volumes and capacities. In further slides, we are going to discuss about the lungs volumes and capacities one by one. Dead air. The volume of the air that is not available for gas exchange with the blood is known as dead air. The total dead space is less than 30% of the total volume. Tidal volume. Tidal volume is the volume of gas inspired or expired during each respiration cycle. It is the resting tidal volume between the inspiratory and expiratory level. The tidal volume is about 500 ml for the normal adult. Inspiratory reserve volume IRV. It is the extra volume that can be inspired above the tidal volume. The normal value of IRV is about 3000 ml. Expiratory reserve volume ERV. It is the extra volume that can be expired by a forceful expiration at the end of the tidal volume. The normal value of the ERV is about 1100 ml. Residual volume RV. It is the volume of air remaining in the lungs despite forceful expiration. Its normal value is about 1200 ml. Inspiratory capacity IC. It is the maximum volume of air that can be inspired after reaching the end of the expiratory level. The normal value of inspiratory capacity is about 3500 ml. That is, the inspiratory capacity is equal to TV plus IRV, that is tidal volume plus inspiratory reserve volume. Functional residual capacity FRC is the volume of air remaining in the lungs at the end of expiratory level. FRC is equal to residual volume plus expiratory reserve volume, or FRC can also be represented in terms of total lung capacity minus inspiratory capacity. FRC can be regarded as the baseline from which other volumes and capacities are specified. The normal value of FRC is about 2300 ml. Vital capacity VC. It is the maximum volume of gas that can be expelled from the lungs by forceful expiration after maximum inspiration. It is actually the difference in the volume of the maximum inspiration and the residual volume. So, vital capacity can be represented as IRV plus TV plus ERV. The normal value of vital capacity is about 4600 ml. Total lung capacity TLC is the amount of gas contained in the lungs at the end of maximum inspiration. So, TLC is equal to tidal volume plus inspiratory reserve volume plus expiratory reserve volume plus residual volume. The normal value of TLC is about 5800 ml. Let us examine the lungs function with the help of the instrument spirometer. The most common, accessible and efficient method of measuring lung functions is by means of spirometer. Here is a representation of spirometer. Its purpose is to diagnose obstructive diseases of the respiratory system. It produces a diagram of the volume of air expired in a given time, that is liter per minute. Spirometer shows the rate at which air is expelled from the lungs. It measures the total lung capacity up to the residual volume. This test does not show the rate at which oxygen is absorbed. If the airways are blocked, the rate of the airflow of the lungs decreases. This will show on the diagram and thus indicate that there is a problem in the airways. The most common obstruction stems from excessive phlegm or from swelling of the inner wall of the airways. The patient is asked to inhale as deeply as possible. The patient is asked to exhale strenuously into the spirometer. The patient is asked to continue to expel air for a few seconds despite the strong urge to breathe air. The test is repeated twice or 3 times. Thank you very much for listening this lecture. If you like this lecture means kindly share this video to your friends. For further updates, kindly subscribe my channel. Thank you.